Okay, everyone, so thank you to be there. We are beginning now. Okay, so just a quick reminder of the Neuro'binar program. This is a 1-hour web conference in two steps. Today we have 2 PhD students which will present their thesis in 10 minutes. And then there will be a 20-minute researcher with today Maxime. So just to present, we have Maxime Guye, which will present his subject on ultra-high field MRI in clinical neuroscience. And to begin, there will be Zeinab Hamze, which will present the study of pathophysiological mechanism of 2 forms of Charcot-Marie-Tooth disease, and Cassandre Vielle, which will present her thesis on influence of peers on cocaine use and abuse. So here we go. Zeinab, it's up to you to begin.
Thank you. So good, you can see my slideshow. Perfect. So yes. To start, good evening, everyone. To start with, I'm honored to be a part of this Neuro'binar today. To present myself, my name is Zeinab Hamze, and I am from the team of Neurobiologie Translationnelle at MMG, and I'm doing my project under the supervision of Dr. Valérie Delag. And my presentation today is entitled Study the Pathophysiological Mechanisms of 2 forms of Charcot-Marie-Tooth. So to start with, the major activities done by the peripheral nervous system is to connect the central nervous system to organs. In addition to that, it carries sensory and motor information from to the central nervous system. One of the most important phenomena that occurs in the nervous system is myelination, that increases the speed of nerve conduction. It is also a source of plasticity in a neuronal circuit that is crucial for proper timing and function. As we know that in the central nervous system, myelination is done by oligodendrocyte. However, in the peripheral nervous system, it is done by Schwann cells. So any disruption in the peripheral nervous system in the communication between Schwann cells and the axon, it will lead to something we call inherited peripheral neuropathies, including Charcot-Marie-Tooth disease. So Charcot-Marie-Tooth disease is one of the most common ITN. It affects 1 in 2,500 per person. It is characterized with a motor and sensory neuron dysfunction. So some of the clinical features that are repetitive in these patients is progressive distal muscular weakness and atrophy that begins at the lower limb and progressively and slowly progress to the upper limb. In addition to that, we have some feet and hand deformation, cramps, scoliosis, variable loss of sensitivity, hypo or areflexia. So in my studies, I use in vitro models. As the first one is to study the axonal morphology and function. To do so, we need motor neurons that are derived from human-induced pluripotent stem cells using a 30 days differentiated protocol that will lead finally to motor neurons that we assess usually using immunostaining with the specific antibodies. Also, we care into studying myelination that I mentioned earlier using a co-culture protocol based on plating DRG, which is the dorsal root ganglion from embryos, and inducing later on myelination using the ascorbic acid in order to have neuron Schwann cells and sensory neurons. So basically, the aim of my PhD work is to better understand the pathophysiological basis of 2 forms of Charcot-Marie-Tooth. As for the first aim is to study the pathophysiology of a new mutation that has been identified in a PDXK gene in a Lebanese family. In addition to that, I'll be working on another project separately in order to correct some defective parameters that has been identified in these human-induced pluripotent stem cells motor neurons in vitro of patients having a biallelic mutation in another gene, which is the VRK1. So as for my first project, whole exome sequencing was done on the two affected cousins in addition to their mother in order to study segregation, and led to identification of a missense mutation in the PDXK in the PDXK gene. So these are the patients having this mutation. So PDXK encodes for paradoxal kinase. For paradoxal kinase that plays a role in converting inactive B6 vitamins like pyridoxine into PLC. So PLC is an essential cofactor involved in vital pathways, including neurotransmitters production and amino acid biosynthesis. So to start with, we had immortalized lymphoblast cells taken from our patients, and we compared them to controls. As the first result, we found that our protein, as a protein quantification in patients, it was decreased 40% compared to the control. However, at the mRNA level, we did the quantification also, but there is no significant difference. What we are planning to do next is to start by studying the axonal morphology and function of motor neurons by measuring neuron length, number of branches also per cells. In addition to that, we will be looking at the electrophysiological phenomena occurring in these motor neurons and compare them, of course, to the controls. In addition to that, I'll be measuring proteins and mRNA levels in these human-induced pluripotent systems. Also, we are interested into looking at the possible implication of PDXK in myelination. In order to study that, we will be doing a knockout of PDXK using a lentiviral shRNA in a co-culture of these Schwann cells derived in the co-culture that will lead to having the sensory neurons in culture. As for my second project, my project that has been started recently. Our team has published an article in which they have identified a biallelic mutation in VRK1. VRK1 it's a protein that encodes for vaccine-related kinase 1, which is a serine threonine kinase. It is ubiquitous and mainly nuclear. Its role is to regulate schedule bodies by phosphorylation. These schedule bodies plays an important role in splicing. So in this article, they have identified some defective parameters that are found in vitro after doing the human-induced pluripotent stem cells in the motor neuron. They found a decreased VRK1 protein levels. In addition to that, tendency to decrease schedule body size and impaired neural length and branches. So the aim of my work will be to try a pharmaceutical treatment using a proteasomal inhibitor known as MG132 on these human-induced pluripotent stem cells motor neuron in order to restore VRK1 expression and schedule bodies. Also, I'll be trying gene therapy on these motor neurons by inserting a native form of VRK1 in these human-induced pluripotent stem cells using AAV1. So this is basically a general view of my work that I'm planning to be doing during my PhD. And thank you for listening. And I'll be happy to answer questions. And if you have further questions, I'll be here to answer. Thank you.
Thanks, Zeinab. So if you have any questions, you can ask them in the Q&A section or the chat, everything, but we will answer them after Cassandre's presentation. So Cassandre, you can go.
Can you see my screen and hear me correctly? Thanks. So, well, thank you first for the invitation. And well, I'm Cassandre Vielle. I'm doing my thesis under the supervision of Dr. Christelle Bonnet at the INT. And during my thesis, I studied the influence of the social context on cocaine use and abuse, and the role which is played in such processes by the subthalamic nucleus. So first of all, why to study the social context on drug use? Well, as you can see in this schematic representation of substance use disorder, or SUD, not all recreational drug users will develop SUD symptom, but only 15 to 20% of the population. So the question is, what factor may prevent or increase the risk to develop SUD? And most importantly, how to help drug abusers to regain the control over their consumption. Until now, there is no efficient medication to treat cocaine use disorder, and actually addiction research is facing a translational crisis, meaning that what does work in animal model does not work in human. This probably reflects a mismatch between what's going on in animal models and the reality, and that's why we think that it's very important to take into account the environmental influence of drug use. So in our team, we assess the effect of the immediate social context on cocaine use. And in a previous study, as you can see, we showed that rats consume less cocaine in presence of a peer than when alone. So these results are promising, yes. In this study, rats had access to the drug only during 2 hours per day. And it's a bit tricky because it's well known that with a such short access to the drug, rats will take a few number of cocaine injections. Their consumption will be stable over days. And there is very low probability that they develop some SUD symptoms. So the question is, will the peer presence be still efficient after rats lost the control over their consumption? To model such a lot of loss of control, it's pretty easy. We just have to give rats 6 hour access to the drug per day. And as you can see, they will increase their drug consumption. This is what we call the escalation of drug use, and it's visible only here in this graphic, only during the 2 first hours of the session when we compare to the baseline or the recreational access to the drug. So then after this escalation, we assessed the effect of the peer presence. And as you can see, even after the loss of control, rats still consume less cocaine in presence of the peer than when alone. And this decrease is very huge because their level of consumption returned to a baseline or to a recreational level. So interestingly, not all peer presence may modulate similarly the drug intake in rats. Actually, in rats, and it's also true in humans, when the peer is a stranger, the drug consumption will decrease more than when rats are in presence of a familiar peer. So after that, we try to understand how does it work, what is responsible for this modulation of drug intake in presence of a peer. And to do so, we assess the effect of vocal communication on drug use. Actually, rats communicate with each other through the emission of ultrasonic vocalization, or USV, and this USV can be categorized in 2 categories. The 50 kHz USV are expressed in appetitive situations and they are rewarding for the recipient rats, while in contrast, the 22 kHz USV are emitted in stressful situations and they are aversive for the recipient rats. So as you can see, when we played back such positive USV emitted by a stranger rat in black on the graph, we show that, when we play back such positive USV emitted by a stranger rat during the cocaine self-administration, it decreased the drug consumption of the rat. In contrast, the playback of positive USV when they are emitted by the cage mate failed to show such modulation. So this result suggests that the vocal communication play a key role in the influence of social contact and drug use. But now, what are the neural bases of such processes? In our team, we focused on the subthalamic nucleus, which is a little structure in the basal ganglia and which is mainly known to its role on motor function. However, studies from our team already show that electric deep brain stimulation at high frequency may prevent cocaine escalation, and more importantly, it can reverse the cocaine escalation after a period of abstinence. So the STN deep brain stimulation will constitute an interesting treatment for SUD. But is the STN also involved in the social influence of drug use? To answer this question, we first use optogenetic tools to inhibit the STN activity during our escalation experiments. And more specifically, we inhibited the STN activity only after the escalation, so it's the green part here. As you can see, when alone, in comparison with the control blue group, rats with the STN inhibition in red decreased their consumption. So the STN inhibition is sufficient to decrease drug use. In contrast, in presence of a peer, we show similar decrease between groups. So we have no additional effect of the STN modulation and the peer's presence, which suggests strongly that social contact influence is actually underlined by the STN activity. Finally, in the USV experiment, the STN was lesion, so its activity was suppressed during the complete experiment. And we showed no effect of the USV playback on the consumption of this STN lesion rats, independently of the USV emotional contact or the familiarity with the USV emitter. So this result confirmed the key role played by the STN in the influence of social contact on drug use. So finally, this work is only a part, obviously, of my whole disease work, in which I try to unravel this effect of the STN in the influence of social contact and drug use. I study its implication also in food versus cocaine motivation and its involvement in social recognition and reward. And I hope that its work will help to better understand the influence of immediate social context on drug use and to better characterize the role played by the subthalamic in such processes. So thank you very much.
Thanks, Zeinab and Cassandre. So now it's a time for questions. So do not hesitate if you want to ask them directly to raise your hands virtually or ask your question on the Q&A section. Maybe Cassandre or Zeinab, you have a question for each other?
Yep. I've been wondering why did you choose the STN particularly from the brain to study it? So if you can elaborate a bit, why the structure basically?
Well, as I said, the structure is mainly known for its motor role, so it's sure that it can seems a bit weird to choose it, but actually it's not, because a lot of work from our team show that it is strongly involved in the motivation, and notably what is very important is that the subthalamic show a dualist role on the motivation, meaning that when you inhibit its activity, when you use the electric deep brain stimulation, you will have an increase in the motivation for food and sugar, notably, while you will have parallelly the decrease of the consumption for the cocaine. And it's very important, because if we try to target the classical structure which are involved in the reward circuits, what you can have is that you will lose the motivation for the cocaine, but you will parallelly lose the motivation for everything. So it's very a big deal. That's it for me.
Yeah, thank you.
And I also get a question for you in return. Could you please explain a bit more the strategy you want to use to treat the patients? Because I'm clearly not a molecular specialist, and I clearly don't understand how it does work.
We are not going to treat the patients directly. We are working on the human-induced pluripotent stem cells motor neurons. So we follow a protocol to order having finally motor neurons in our plate. And I'll be trying to treat it with the pharmaceutical treatment, which is the MG132. It is a proteasomal inhibitor, so we think that VRK1 in this project is eliminated by the proteasome. So if I block the proteasome using this MG132, I'll be able to restore my VRK1 expression in the motor neurons that are in the plate, not directly in the patient until right now. And for the gene therapy, it's going to be using the AAV6, which I'm going to insert in these motor neurons to replace the native, the VRK1 mutated with the native form, and see if we can also restore the defective parameters that I've explained earlier. Yeah.
Okay, so there is several questions. The first one for Cassandre. Does the sex of rats is important in your experiments?
Well, in my experiment, I only used male rats, actually. It's a very good question. We don't know what's will going on with female. That's something that we will have to do in the future. The fact is that literature seems to indicate that social contact is more rewarding for male than female. So clearly it seems possible that there is an effect of the sex, and it's very important to replicate it with female rats.
Okay, thanks. The next one for you, Zeinab, is DDXK expressed by neurons and glial cells?
Looking at the data we have online, yes, we can say that PDXK, it is expressed in the neurons and the glial cells, but we are going to look further into these motor neurons by doing these experiments to see how much is it expressed, and if this expression is decreased in the patients having the mutation of PDXK. So yes. Thank you.
Okay, thank you. We have more for you, Cassandre. From Mark Bartoli, the number of rats pairing have an effect in the behavior?
The number of rat pairing, you mean the number of sessions that I will perform with a peer?
I don't know. Maybe Mark, do you want to ask your question directly? Yes. Okay. You can raise your hand. Yeah. And here we go.
Okay. It works? Yes.
Yeah.
So thank you, Cassandre, very interesting. And no, I just wondering the number of rats that are pairing of, in fact, the rats that administrate the cocaine. If there was only one rat, 2 rats, 3 rats, and
yeah.
If the number of rats is important for the social context or not?
I think so. We never try with several rats as observer. Actually, what we do is basically we let one rat self-administering the drug, and is in presence of only one peer. But yeah, it could be interesting to do with several peer and see how it works.
Thank you.
You're welcome.
Thank you. We have a question from Anne. Anne, do you want to maybe ask your question directly? Okay, so I will read it. You show that vocal cues can modulate the behavior of rats towards the drugs. What about direct touch between animals?
Well, in our experiment, we didn't actually rats are separated from each other with a wire mesh, so they can have a little bit of touch, which is restricting because there is a wire mesh, but there is it's the best. There is one study which tried with with a plastic wool in separation of the two rats, and they don't find the same result than us. So when there is only the vision, it seems that we don't have the same effect. So probably the touch will play a key role too. Yet we also have an effect when we do only the USV. So there is no touch, nothing which is just present. Actually, I'm a bit tricky with this question because it's a bit tricky for me, this question, because the only experiment that I know where they try to separate the rate by a transparent wall, I don't know if we can say that the vision is still conserved because they use some kind of Wistar rat, something like that, and I'm not sure that this rat can really see actually. With this plastic wall. So I think there is an effect of the touch globally, but I don't think it's really strong because USB does work. So
okay, just the last one quickly from Lydia. Is activation of STN neurons suppressing the influence of stranger peer?
Well, it's not its activation. It's inhibition. It's inhibition, sorry, or lesion, which can seems to suppress effectively the presence of a stranger peer, but only in certain circumstances, meaning only after the loss of control over the cocaine intake and for the USB, but we didn't find this effect when we do the experiment on the recreational access, during which rats with STN lesion actually show a very strong effect of the stranger peer presence. So we still don't know for that. We are still trying to perform experiments to answer this question.
Okay, thank you. So we will go on with Maxime. Oh, I don't know if you can see my screen. I don't know if it's okay. Do you see my screen or not?
Yes, it's okay.
Yeah, it's okay. Okay, thank you. So I will just present Maxime just rapidly. He's a professor of biophysics at the AMU Faculty of Medicine and senior consultant in the Department of Clinical Neurophysiology of the University Hospital. He held a research position in neuroimaging at the University College of London after his residence in Marseille. He is a member of the scientific committee of the French Society of Radiology, the French Society of Clinical Neurophysiology, the French League Against Epilepsy, and the French Foundation for Research Against Epilepsy. He was elected a member of the National Committee for Scientific Research and the president of the French Society for Magnetic Resonance in Biology and Medicine. Maxime has published over 100 peer-reviewed papers in the field of MRI and neurology. His research interests are ultra-high field MRI, combining and comparing multimodal MRI method with electrophysiological recordings and brain connectivity. Here we go. So if it's okay for you, Maxime, you can go.
I share my screen. All right. Should work.
Yes, it's okay.
Thank you for the introduction and for inviting me to talk about ultra-high field MRI in clinical neuroscience. So we talk about where we are currently and where we are heading. And to start, you have an example of a sample of the development we have achieved during the last 6 years in the lab. And I will show some example in more details in a minute. Yeah, maybe to start, a few words on Center for Magnetic Resonance in Biology and Medicine, CRMBM, Centre de Résonance Magnétique Biologique et Médicale in French. That is a lab devoted to the development and application of innovative MR methods for clinical sciences in cardiovascular, musculoskeletal, and central nervous system diseases. And obviously today we'll talk about neuroimaging, so about the work developed in the central nervous system group devoted to human that is headed by Jean-Philippe Rangeva. Well, on the facility, this slide shows both our location in these two sites and our facility. So the first site in the medical school, devoted to preclinical imaging. We have 3 scanners, and in the medical site, this site is called CMRM because it's also a clinical department belonging to the hospital also, where we have 3 scanners, 1.5, 1.3T, and recently a new 7 Tesla system that has been changed a few months ago, and the new system is approved for clinical use in neuro and MSK indication, in some neuro and MSK indication. So what can we gain with ultra-high field? By increasing the strength of the magnetic field, the B0, we increase more than linearly the signal to noise ratio. So this is beneficial to the spatial resolution, to the temporal resolution if we use a dynamic acquisition. It is also beneficial to the nuclei other than proton, higher sensitivity, for instance, for sodium or phosphorus, which has usually a weaker signal in MRI. Also an increase of the contrast to noise ratio, particularly an increase in susceptibility, and that will be particularly important for the T2 star weighted imaging, the susceptibility weighted imaging that can be also quantified, so using the QSM technique, the quantitative susceptibility mapping. And also, obviously, this will benefit also to the bold signal with a highly increased sensitivity in fMRI. We also have an increase of the T1, so the longitudinal relaxation time, that will benefit to magnetic resonance angiography and also to perfusion using arterial spin leveling. Also an increase of the chemical shift, so a higher spectral resolution for spectroscopy, both proton spectroscopy or phosphorus spectroscopy, and also a better definition using the CES techniques of the chemical exchange saturation transfer technique that can be used more easily at higher field, for instance, 7 Tesla. And it is particularly the increase of spatial resolution, the increase of susceptibility, and this increased T1 that benefits to the MR angiography that have led to the approval of the 7 Tesla MR scanner as a medical device in neuro and MSK indications. So what do we gain? In terms of spatial resolution, you have here an example of the high resolution we can achieve. So a better definition of the subcortical architecture. It is the case, as you can see here in the T1 weighted template. You can see here the resolution where you see nicely the subnuclei of the thalamus. We can do some volumetry, T1 measurements, so quantitative measures. And recently, Gilles Brun and Arnaud Trotter particularly have developed in the lab a high resolution atlas of the thalamus and the basal ganglia. As you can see here, it's also beneficial to the high resolution to the better definition of the brainstem and the numerous nuclei you have in the brainstem. Also better for the cortical architecture, and the 7 Tesla can bring new insights into the better cyto and myeloarchitectonical organization of the cortex. As you can see here, for example, in the T1 weighted imaging, you can see the T1 measurements, the T1 value that differs from the depth compared to the surface of the cortex. And you can see here in the Marquez group an interesting study showing the different value of T1, T2 star or susceptibility in the different depths of the cortex. Also the possibility to have the cortical fiber orientation, for instance, in this paper showing nicely some tractographic measurements, the cortical orientation of the fibers within the cortical regions. So this, thanks to this higher resolution, the 7 Tesla structural MRI may impact the clinical management, so have a real added value in the clinic by modifying eventually the patient outcome in several indications. First, epilepsy, you have here the list. Epilepsy, particularly in refractory focal epilepsy, multiple sclerosis, in tumors, particularly some gliomas and pituitary adenomas, in some neurodegenerative disease, particularly Parkinson's disease, and it is increasingly used in deep brain stimulation, not only for Parkinson's disease, and also a good indication in cerebrovascular diseases affecting the small vessels, showing also some micro infarcts or microbleeds, and can also benefit to a better definition of malformations such as aneurysm, for instance. So what can we expect next? So beyond these structural MRI abnormalities, we can expect not only the better structural definition of lesions, but also better mechanism assessments of the disease. And to enter a bit more into details, I would like to take the two first examples, epilepsy and multiple sclerosis, which are two diseases we are particularly interested in, and to show you some example on how to use this information right now in the clinic and what we can expect next in order to improve the management, to improve a better understanding of the pathophysiological mechanisms. So let's start with epilepsy. So as you know, it's a very common disease. Up to 1% of the population is involved. It's around 6 million people in Europe. A huge burden at the individual and societal level. Individually, it increases significantly the morbidity, also the mortality, and at the societal level, it costs around 20 billion a year in Europe. And 30% of patients are refractory to drugs, so drug resistant. And in the patient in whom you can define in the brain the regions responsible for the seizure, the only curative solution is to remove this region surgically. And so this is a clinical context. So in drug-resistant focal epilepsy, for the presurgical assessment, the presurgical evaluation, it is well demonstrated that detecting, characterizing, and accurately delineating a lesion can improve surgical outcome, can avoid or at least guide invasive recording. And between 20 to 40% of patients are still have still a normal appearing MRI at 1.5 or 3T. And so in this topic, there is a clear demonstration of the added value of 7T, since a 7 Tesla MRI can reveal a lesion in about 25% of patients with normal appearing MRI at lower field, particularly with focal cortical dysplasia, that is a well-known epileptogenic lesion. It can not only better depict, but also better characterize and delineate lesions, and also correct for ambiguous finding. And this is very important from a clinical point of view. Particularly using these sequences, I will say a few words on that later. With a particular interest in malformation of cortical developments, as I said, particularly focal cortical dysplasia. And we have this impression on site that is well also demonstrated by others, that 7 Tesla in any case is useful in patients that will be operated on. So either with normal appearing MRI at lower field, or even in patients in whom we have some lesion at lower field that will be better characterized with the 7T. Here is an example. Focal cortical dysplasia, a patient presenting with frontal seizures, normal MRI at 3T, and at 7T, you can see this increased signal in T2, this thickening of these cortical regions in the anterior part of the cingulate gyrus. In this T1 weighted sequence, MP2H, you can appreciate here the resolution. That also provides a T1 mapping, so a quantification of the T1, and also because it uses 2 inversion time, the first inversion offers also a very interesting contrast. As you can see here, you have a black line that perfectly delineates the border between the gray and the white matter. And in that case, and it is the case in focal cortical dysplasia, you can see this disruption showing the dysplasia responsible for seizures. Also another sequence, the flows, it's a double inversion recovery. So you use 2 inversion to actually suppress the signal from the CSF and from the white matter. So you end up with basically a quite pure gray matter signal, as you can see here. And in that sequence, you can appreciate here what we call the transmuscle sign that connect the focal cortical dysplasia to the ventricle here. That is pathognomonic of focal cortical dysplasia. We have now the possibility to generate this sequence from the MP2H, so we can get rid of this flow sequence thanks to a recent work in the lab. Also another kind of lesion, the hippocampal sclerosis, that is also a very common lesion. And what is interesting with the 7T is that we can define not only the sclerosis of the hippocampus, but specific atrophy of subfield of the hippocampus. This increased spatial resolution obviously can be also very interesting for Alzheimer's disease, of course. As you can see here, with this nice resolution, you can see the digitation of the head of the hippocampus. In this paper, you have all the substructure of the hippocampus you can see at 7 Tesla. Also, venography is here using susceptibility imaging, also using MR angiography, the vascularization, all the small arteries going to the hippocampus. And in this paper, you can see the quantification of the atrophy of the different subfield of the hippocampus that can characterize mainly 2 types of hippocampal sclerosis that are usually depicted only in pathology and which can lead to different semiology in the patients. So thanks to this clear indication, we have gathered together with Anja van der Kolk and Giske Ofheim, what we call a 7 Tesla epilepsy task force, gathering 21 centers. And we published a consensus recommendation for the use of 7 Tesla MRI in clinical practice in this indication in epilepsy. And we think this is what we have to do in all the indication that have been proposed so far. So we have defined the indication, all the patient selection preparation, the acquisition protocols, the setup, and the guidelines also for interpretation. We benefit from the multiparametric characterization of lesion, obviously, thanks to the increased contrast to noise ratio. And you can see here in this lesion, all the different contrasts we can get that help to better define, to better characterize the lesion. For instance, in this patient with a high signal in quantitative susceptibility mapping, in a patient presenting with an angiocentric glioma that has been operated and is seizure-free. It's also the case for the first patient. And thanks to the quantification, we think we can, in the future, maybe using some artificial intelligence algorithm, moving towards an in vivo phenotyping of epileptogenic lesions. We can also better guide the surgery, so improve the surgical planning, both for structural connectivity using higher resolution diffusion MRI or higher sensitivity of fMRI. But here you have an example of tractography using a high resolution diffusion MRI sequence, and you can see the relationship between this tiny focal cortical dysplasia here in the depth of this sulcus, like you can appreciate here, and the optic radiations depicted in this image, or the middle longitudinal fasciculus, for example, instance here, with the lesion here. We can also better guide the invasive recording and improve electrode localization and implantation for surgery. You have here an example where we show the relationship between the 3 leads of an electrode depth electrodes that are the most involved by epileptogenicity, that correspond well to the location of, again, this very tiny focal cortical dysplasia in the depth of this sulcus. So we can better define epileptogenic lesion, but the epileptogenic network, the network responsible for seizures, can extend beyond this lesion. So assessing more comprehensively neuronal glial impairments and network alterations in particular is particularly crucial. And in that topic, sodium homeostasis, quantified by sodium MRI, is a good candidate. We have demonstrated a few years ago a sodium accumulation in epileptic regions using 3T MRI. Now, at 7 Tesla, thanks to the work of the group of Wafa Zahraoui in the team, we use a multiparametric approach thanks to the higher signal. Taking advantage of the higher signal, we use a B exponential fit of the decrease of the sodium signal, and we extract from this the short component here that is more related to a restricted pool of sodium, so mainly weighted by the intracellular sodium, compared to the long component that is more related to the mobile pool, so mainly weighted by extracellular. And for instance, we have computed what we call the HEF, signal fraction that represent this short fraction over the whole signal of sodium. And Michael Azilino, who is doing his PhD in the lab in collaboration with the Institute of Neuroscience des Systèmes with Victor Girsa, has compared different, all these parameters from sodium MRI with SEEG, so depth electrodes recording. And for instance, for the HEF, he demonstrated nicely that this HEF was increased. So there was an increased HEF in the epileptogenic zone compared to the same regions taken with sodium MRI in the healthy controls, and also an significantly increased F in the epileptogenic zone compared to the propagation zone of the non-involved regions in the patients. And now Michael is using this information, this parameter, to better inform some computational models developed by Victor Girsa's team in some models that are used and that are called virtual epileptic patients that reproduce the behavior of the seizures at the individual level in individual patients. We can also use other information, for instance, glutamate increase that we can get from CEST imaging, and acetyl aspartate decrease that is related to mitochondrial deficits that we can get from spectroscopy, proton spectroscopy, and also some connectivity measurements from diffusion or resting state fMRI. Okay, so the second example, I'll try to speed up a bit. It's multiple sclerosis, again, a bit of clinical context. It is the first non-traumatic cause of disability in young adults. It's an immune-mediated inflammatory disease affecting the central nervous system, causing lesion of demyelination, following mainly relapsing remitting course, and then eventually, in some cases, going towards disability due to neurodegeneration. So what do we need? So the clinical context, first, the positive diagnosis is based on dissemination in time and space of the lesion in the central nervous system. So we need to depict all the lesions. An early and sustained treatment reduces the risk of long-term disability in the relapsing remitting form of MS. And the target for the treatment is no evidence of disease activity. So we need to quantify the activity of the lesions. And we still don't know how to predict neurodegeneration. It's the case for multiple sclerosis, but it's also the case for all the neurodegenerative diseases, actually. So here are the demonstrated added value. It can help with the differential diagnosis of brain lesions by showing a vein in the center of this lesion that differentiate MS lesion from other diseases that can cause the same kind of lesion in the MRI. It better also characterize and depict the lesion in the gray matter. This is very important in the cortex, but also in the thalamus, in the subtemporal area, and also in the spinal cord. And this is very important. I'll show you some example. And it also brings some clues for better characterization of lesion activity, thanks to different parameters. Some example, so we assess the lesion activity using a multiparametric approach. Again, you can see here what we can get from the MP2 range. In the T1 map, you see here this little lesion, a pure cortical lesion, you can see here, with a slightly increased T1, not that much. You can see here the nice delineation between the white and the gray matter. So this lesion is purely cortical, and no change in flows or in QSM and susceptibility measurements. And in this lesion, this is different. As you can see, this is a subcortical lesion with a higher T1 in the lesion. Also, this paramagnetic rim that sign a high activity of the lesion that can evolve toward the progression. 7 Tesla is much better for the spinal cord. This is the work of Virginie Callot's group in our lab in collaboration with the clinicians, obviously, as usual. You can see here, for instance, this lesion that is visible only in T2, not at 3T. Again here, 3T, 70 here, that allows for a delineation, clear delineation of the lesion, segmentation of the lesion that is useful to follow the patients. So Virginie has developed a multiparametric approach, a quantitative approach of the spinal cord, and you have here all the papers we have published in the lab around the spinal cord studies at ultra-high field. And you can appreciate what we can have in terms of anatomical imaging. Again, in order to better assess lesion activity, but also the degeneration, sodium MRI can have a role. Wafa Zahraoui and others in the lab have demonstrated already a link between the sodium accumulation and disability. More recently, in this multiparametric approach, we demonstrated that this F, and this signal fraction F in lesion and in normal appearing white matter could differentiate patients with complete recovery from patients with partial recovery. So again, an information on the neurodegeneration. This is for metabolism, and I will maybe finish with this. I didn't talk about neurodegenerative disease per se, even if we have some degenerative processes in MS, but I wanted just to share with you a recent paper we've just published with Stéphane Grimaldi, who is doing his PhD with me and Alexandre Eusebio, showing sodium accumulation in the substantia nigra in Parkinson's disease. So we selected different regions of interest, and the increase of sodium concentration was significant only in the substantia nigra in Parkinson's disease. And we think this could be very interesting to differentiate Parkinson's disease, pure Parkinson's disease, from other Parkinsonian syndromes, for instance. So I didn't touch upon cognitive research, but obviously for me, 7 Tesla is a paradigm shift in neuroscience. You can better study anatomical and functional organization of the cortex and subcortical regions. You saw some example. I didn't talk at all about the mesoscopic organization of information flow and connectivity that you can get with fMRI, particularly laminar fMRI. We have some studies starting in that topic. And also the possibility with ionic and metabolic changes that we can measure to better understand brain activity and dysfunction. I will stop here. I have one paper, but I don't have time maybe to discuss it. It's a dynamic sodium MRI published recently by Marc Bider and Jean-Philippe Rangeva that have been compared to fMRI, but we can discuss this if we like and if we have time. And to finish, I would like to thank all the people involved in the 7 Tesla in the neuroimaging part, also the clinicians who are working with us, and the Institut Neurosciences des Systèmes with a big collaboration, particularly in the EPINOV project and the HBP project, the Human Brain Project, the European project, where we use many of the parameters from the 70 to better inform all the computational models that are developed in this project. Okay, with this, I have finished. Thank you for your attention. Thank you very much.
Thanks a lot, Maxime. So we have already one question, two. So the first one is from Anne Cavunijas. You can read it. Do you see it, or I read it for you?
All right, I try.
Oh, maybe Anne, okay, Anne, maybe you can ask your question directly.
Okay, okay. Well, I was in the chat window, but there is another window which is the Q&A, and okay, so let me have a look. Head movement. Okay, the movement. Yes, right. How do you manage this, especially for clinical? Second question, have you already done functional? Okay, so I will start by the last one. Done functional MR acquisition at the spinal cord level? The answer is no. To my knowledge, there is only one group that has done so. I have to be honest, I am not totally convinced by this, but this is definitely something we will try. That's for sure. For the movement, obviously, by increasing the resolution, it's more. We will be. We can have more problem with the movement. To be honest, in our experiment, we don't have that much movement. That can affect the quantification, obviously, but so the correction are we don't use the inline correction, the online correction that can be done with the scope device, for instance. So we basically use both. I mean, fixation of the head during the experiment and some post-processing processes. Other than that, we don't have specific tips for that. Yeah, no. But to be honest, we are surprised. We can interpret most of the patients, most of the scanning patients. I don't know if I answered correctly, but it's true that we had many young patients with multiple sclerosis or epilepsy patients. We have scanned many patients with Parkinson now. Without that problem, obviously, we select patients with less movements, but no, we don't have too many troubles. Should I follow the rest? Okay.
Yeah, you can go to the question. Sorry, you can go to the question of Laura.
Okay, overdiagnosis. Okay, the question of overdiagnosis of really small lesion when resolution increases. That's also a very good question. That is true. The main example is the microbleeds. We see many microbleeds. So basically at the beginning we thought, well, so many microbleeds doesn't mean anything. And then we realized that in patients with Alzheimer's disease that have been scanned in postmortem, they had many microbleeds. And so then the microbleeds became a risk of neurodegenerative diseases. And it is now considered as a potential risk. Same for perivascular spaces. For instance, we identify more easily perivascular spaces, and we are discovering some perivascular spaces increases in some diseases, such as epilepsy, for instance. So it's difficult. Yes, it's true. We find some lesion, some small lesion, incidental lesion, and it's difficult to deal with. But on the other hand, we also discovered things thanks to these small lesions. So it's an issue. It's true, but it's also an advantage, clearly.
To follow, you have one from Fabrice. Can you clarify the spatial resolution and the temporal resolution you can now get to?
Okay, so spatial resolution, I showed in my example, the usual spatial resolution we get in a protocol that is used for the clinic. For instance, for MP2 rate, so the T1 weighted sequence is a 0.6 millimeter cube resolution, 600 micron, in 10 minutes. Same for QSM, and the same for the flows, and for the T2 also. So we end up with an acquisition that lasts a bit more than 30 minutes, or which within 30 minutes, let's say, just for the structural MRI, with a resolution of 600 micron cube. And for the temporal resolution, it depends. If we for fMRI, we use the HCP sequence, that is for the whole brain. So for the resting state, you can, and the TR is 1 second. You can go faster, but then you have to use a compromise between the temporal and the spatial resolution. We have tried, and we went below 1 second, but still, you are still suffering from the resolution of the bold signal anyway. And the last is the DBS.
Yes, the last one from Lydia. What are the main expectations for the use of 70 regarding the treatment by DBS?
Yes, so with regard to that, well, the main goal is to better localize the targets from the images. The clinicians are still using electrophysiology, of course, but for the neurosurgeon, it seems that it's better to have more information from the anatomy. So we can see some targets that are invisible at 3T. So this is how it is used. I don't think it will replace the clinician and electrophysiology, not for now, for sure. Even if we have some nice example of stimulation simultaneous to fMRI from Toronto. I don't know, but yeah. These are the expectation, a better anatomical definition of the targets. I hope I answered to the question. I'm not an expert on DBS at all.
Okay, thank you, Maxime. So I think there is no more question. So thanks everyone. Thanks to Cassandre, Zeinab, and you, Maxime, for your time. And we invite you. I was just sharing my screen. Okay.
By the way, the 70 is open to everyone. Okay, so it's an open platform. So if we have projects, you are more than welcome.
Great. Thanks, everyone. And we will see you on December 14th at 1 p.m. with Maxime Guye from INT, Guillaume Frébourg from CRMBM, and Mostafa Seblani from the ISM. And again, thanks, everyone. Have a good afternoon.
Thank you,